Tissue engineering, 3D printing human arteries and hearts. So the human heart is really an amazing organ, right? It has this soft complexity. If we look at this video, we see it beating, right? It pumps blood throughout the body. It starts beating 3 weeks after conception and doesn't really stop until the day we die. That's really amazing for anything to last that long and to function with that kind of redundancy and efficacy for that long. It pumps the blood, is electrically synchronized. It has some really amazing properties. It operates for billions of cycles, right? Your car has to have the oil changed after 3,000 miles, a couple months, right? This lasts 40 years, even if you abuse your body, and for most of us, a lot longer, right? It's chemically powered, so the energy stored in sugar is 2 orders of magnitude higher energy density than what you have in gasoline. It's electrically synchronized, which if we watch that video, controls where the heart beats and when it beats. That maximizes the amount of blood that leaves the heart. It's composed of over 100 billion cells, okay, of all different types. Cardiomyocytes, which I'll talk a lot about today, these are the heart muscle cells. These are the cells that beat and contract and generate force. But there's also a conduction system, fibroblasts that help build the heart, endothelial cells that surround your blood vessels and help maintain blood flow throughout the body, and neurons that help regulate your heartbeat. The problem with the heart, and the reason that we are interested in trying to rebuild it and engineer it, is that it does not regenerate. And for this reason, heart disease is the #1 cause of death in the first world. All right, it doesn't really matter where you are, it is the #1 cause. So in the third world, more basic issues like the cleanliness of water are a bigger concern. But in the US, in Asia, in Japan, and in all countries that are developing, this is the major concern. So the question we're trying to address is, how do we fix the heart? You know, what are our options? The heart muscle, to repair it, needs to be replaced. So here are a couple different strategies that one can employ to do that. The first is cell sheets, right? So the cell sheets are basically layers of heart muscle cells that you can stack and form a tissue. And what we're trying to do is actually repair a damaged part of the heart. So here we show a dark region. That would be something from a myocardial infarct or an MI. That's what a heart attack is. That's where a blood vessel that actually feeds blood to the heart becomes blocked, and anything downstream of that blockage is starved for blood and that muscle dies. Another option is just to inject cells directly in a syringe. That's cell injection. A third option is to embed cells in kind of a gel and paste that onto the heart. A fourth option would be cells seated onto what we call a nano or microstructured scaffold. This is an engineered material that we put cells on. It grows into a tissue, and we place that on the heart. The fifth option is to seed cells onto what we call an extracellular matrix scaffold, or ECM scaffold. So none of those options really work, and part of the problem is that the heart is really an amazingly complex biological machine with a structure and function that scales from the nanometer level on up to the kind of the macroscopic level, okay? What that means is that every heartbeat, right, we can think about the heart contracting, right, or our muscles flexing, right, but every heartbeat is actually powered by molecular motors, all right? Molecular motors are only a few nanometers in size, and they generate piconewtons amounts of force. A piconewton is 10 to the minus 12 newtons. So that's billions of times smaller than the force you would generate, say, out of a muscle contraction of your arm. Now, these are built into larger scale structures called a sarcomere. A sarcomere is kind of like the contractile unit of a muscle cell. It's only a few microns in size, even smaller than the width of a human hair. And these are built into fibrils that span the length of your heart muscle cells, shown here. And these heart muscle cells kind of almost form like a brick wall. If you think about the bricks in a standard wall, you can think of those as the heart muscle cells. They form sheets, and these sheets wrap around the ventricle. You can think about the ventricle like a roll of toilet paper, right? You've got the hollow chamber inside with paper wrapped around it. Imagine that's muscle. Okay, that contracts, all right, it decreases the size of the ventricle and pumps out blood. Okay, so trying to engineer anything this complex from the nanometer scale on up to the macro scale is so far beyond current technology that we really have no chance of ever trying to engineer something that complex from the bottom up in our lifetime. But we don't have to, right? Every one of us is a walking example that this is possible, right? That the biology knows how to build these complex structures and make it highly functional. So what we do is we figure out where we can interface with the system, provide information into the biology that can help guide the cells and the living system to do a lot of the work for us, okay? So what we have at the micro and nano scale, which is really at the size scale of the cell, is we engineer surfaces and interfaces in 2 dimensions and 3 dimensions to do that. That essentially drives the cell to build everything down to the nanoscale for us. And then from the larger scale, we use 3D bioprinting of microstructured scaffolds to build larger things up to the size of a whole organ. And that's how we go about doing this. But how do we build? Like, how do we actually make that happen? Okay, so traditionally, manufacturing, throughout the world has been based on what we call subtractive manufacturing. That's essentially removing material, okay? So let's look at true biological additive manufacturing. So not current 3D printing we use in industry, but what does biology, living systems do? So the embryo is really the best example of biological additive manufacturing. It starts out with one cell, and then those cells divide, and they build their extracellular matrix around them until at the end of development you have a full functioning adult organism, or a juvenile organism, but it looks just like the adult. Here on the right, an example of a zebrafish egg with 2 cells, shown here, on top of a yolk sac. When we look at what happens, these 2 cells divide, and they divide again until they form thousands of cells, which then spread and migrate around that yolk sac. As they're doing this, they're secreting extracellular matrix, assembling them into fibers, building that around them, and differentiating into different kinds of cells, and forming the different parts of the animal. All right, so what you'll see here is this animal spreading out. The backbone is going to form along here. This will spin around in a second. And what you'll see is the eyes developing up here. The tail will be developing down here on the bottom. You'll start to see somites, which are how the different internal organs develop. And eventually you'll start to see this guy squirm around, okay? And that's because the skeletal muscle has now formed and it's just starting to contract, okay? And this happens over a period of hours in the zebrafish. Obviously, it takes us a little bit longer as humans, but we end up very close to the same place at the end. A vertebrate, fully functional, that essentially has the main features of the adult, okay? That's the place we would like to end up. Right now, we're far from that, but we're building the tools and technologies, hopefully, to get us there. But getting back to this idea of the extracellular matrix, this ECM, this biomaterial of the body, what I really want to hit home on is that the extracellular matrix is information. It's different than other kinds of materials like metals and plastics because it can provide cells instructive cues on what to do, how to organize, how to form tissue. And that's really a unique aspect. So again, it is more than a material, okay? Cells can use it. It has physical, chemical, and mechanical properties that is integral to tissue form and function. There is no us without extracellular matrix, and therefore it is really an ideal material to use when we try to rebuild tissue. And so what my lab has done is develop techniques that we call bottom-up engineering of the extracellular matrix. So trying to take this idea of how an embryo, a single cell initially, secretes and builds the extracellular matrix around it, right, eventually building a full tissue, right, that's from the bottom up, starting with one cell. Can we do a similar thing, taking the same extracellular matrix proteins, but in an engineered system and build that from the bottom up? If we can do that, then what we hope to do is take single molecules of proteins, these extracellular matrix proteins, just like cells do, and build complex 3D networks that are specific, right, in structure and function. That means specific to the organ that we're trying to engineer. Okay, and if we could do that, then we hope to access synthetic stages of development, right, and accelerate traditional morphogenetic programs. I know that's a mouthful, but what does that mean, okay? Synthetic development means, right, it takes us 18 years to get to adult form, and if we want to replace a tissue in someone who's 60, it took 60 years to get to that tissue, right? We don't have 60 years to wait for any application. I don't even know what the cost of that would be, but it's astronomical, okay? So we want to take principles from development in a synthetic system and accelerate that, okay? Make it happen faster, okay? And that's also what we mean by accelerating traditional morphogenesis. Morphogenesis is just the formation of a tissue or an organ. So we know that the ECM, or the extracellular matrix, can guide muscle formation, and we have good evidence that this definitely works in 2 dimensions. 2 dimensions just means on a flat surface, okay? And the way we know that is from something called microcontact printing. Simply put, this borrows technology that is used to make microchips, okay? And instead of making microchips, we make rubber stamps with micro or nanoscale features on the rubber stamp. So here's an example of what that looks like. So here's a surface where we've engineered linear strands of cardiac muscle. The reason we do this is that the muscle in your heart is highly aligned. It's highly aligned because if all the cells face the same direction, it maximizes force generation in that direction. It's the most efficient structure and the most powerful structure. And that's why we want to pattern cells in 2D. If you don't do this, cells grow in every direction randomly and it becomes highly efficient and essentially mimics disease tissue and not healthy tissue. So how do we transition from a 2D system and move to a 3D system? So the simplest approach is actually just allowing a 2D system to bend in 3D, right? So you can think about a film that can contract and move. And so we've developed a technology in the past called muscular thin films that allows us to do this. Okay, it's kind of similar to a fruit roll-up for those of you that may be familiar, right? So a fruit roll-up is really just a plastic film, and then you have some fruit on one side and a thin layer. And I won't really say it's fruit, but it's fruit-like, okay? And it is very similar to what we have with a muscular thin film. Imagine that fruit now is a thin layer of engineered muscle, and you have an elastic film. So you have a two-layer film, okay? So when that muscle contracts, it bends to the side of the muscle, if that makes sense. So here's some examples from a paper we published a few years ago while I was a postdoc at Harvard, taking this muscular thin film technology, okay? Here we have a rolled strip. So this is a long strip, okay, with muscle on the inner concave surface. You can kind of see the muscle strands lined along the length here. And when they contract, it decreases in diameter. All right, this is actually very similar in the way that a ventricle decreases its inner diameter during a heartbeat. Here is a second example with a gripper, okay, where we open and close the gripper by controlling how fast we stimulate the muscle. When we stimulate it very fast, it stays shut. Okay, when we stop stimulating it, it's open, all right? This is similar to the way that we can open and close our hands. This is a myopod, so this is a little bit harder to maybe visualize, but this is a piece of this film walking along the bottom of a petri dish where we're just stimulating it to contract, okay? So we call these soft robotic devices. They're ways that we can build muscle actuators that actually can start to do work, all right, and can mimic true organism function. Here's an example of a piece of heart muscle, but not just any heart muscle, it's right ventricular heart muscle in the mouse, right? So this was the first time anyone demonstrated that you could engineer not just heart muscle, but a heart muscle specific to a subregion of the heart, because the different parts of the heart are actually quite different in their electrical properties. So 3D bioprinting. So this is really our attempt, and not just us, but many other groups, to combine material science, okay, additive and advanced manufacturing technologies, as well as a good understanding of cell biology to really reinvent how life is built, okay? Not, new life, but how we can build living systems, right, to treat and address issues with human disease and health, okay? The challenge of soft materials, and that's essentially what we are, right? We're made up of bone, but the rest of us is very, very soft. And so the challenge there is that soft hydrogels, and hydrogels are things like collagen, fibrin, which is the material of blood clots, they don't have the mechanical strength to be printed in air, okay? So you can imagine like a block of Jell-O, right? It's very kind of soft and jiggly, right? If you have a solid block of Jell-O, okay, it can kind of hold its own shape, right? But imagine if you start to put holes in there, you start to build any sort of complex structure, it'll kind of deform under its own weight. And the problem that's an issue with 3D printing is that when you lay down one layer and you come back to put down the next layer, that first layer better be where you put it. If it's moved, then you can't build a layer on top of it, okay? And that's really the challenge with soft materials. If we use the support material, we need to be able to remove that support material when we're done, because probably that support material does not belong there in our final printed tissue. And then uniquely to bioprinting is we need to do this all in a system where we can keep the cells alive, which means a very tight control on being in an aqueous environment, a water environment, not air. Control the pH at about 7.4, that's the pH in our body, the right ionic or salt concentration, and then temperature and sterility. Sterility is really a unique aspect of bioprinting. We can't have any sort of foreign bacteria, viruses, mold, fungus around, or they'll destroy the system we're trying to build. So the technology we've specifically built to do this is called FRESH. So FRESH stands for Free-Form Reversible Embedding of Suspended Hydrogels, okay? And what we can do is we can 3D bioprint tissue engineering scaffolds in complex biomimetic geometries, okay? So here's an example of how that happens. All right, so what we do is we take a syringe here. This is from our 3D printer. Okay, we drop a syringe into a support bath, okay, and we essentially print a gel inside of another gel, okay? So you can kind of think about this like the fruit that might be hanging out in a Jell-O mold, okay, right? Jell-O, right, can provide a very good support for materials inside of it, and that's essentially what we're trying to achieve here. But we actually started this research using something called a MakerBot. So a MakerBot is a consumer-grade, open-source-based 3D printer, only costs a few $1,000s. You can buy variants of this now at Staples or Office Max or Best Buy. But what it does is it essentially provides a very low-cost system for having three-dimensional control of depositing material, which is what we want to do in 3D printing. But basically what we have here is we have a Petri dish, okay? We have a syringe coming down that's attached to the 3D printer. It's going to deposit material. And inside this Petri dish, this translucent material is our support bath. All right, so we're going to print, okay, inside the support bath, and when we're done with the print, we're going to dissolve the support bath away, and that will release what we printed. So here we are printing the letter CMU. Now these letters have been dyed black just to help with visualization, but we're going to print them layer by layer. Okay, you can see the stage moving down, right? That's printing the different layers. And when we're done, we're heating the platform that this Petri dish is on to 37 degrees C, and it melts the gelatin, and these letters float up, right? So now what we can do is take this basic idea, and instead of printing the letters CMU, which is great for school pride, but it's not going to help anyone treat disease, okay, is to now move on and take medical imaging data, okay, like an MRI data, okay, use that as a way to print patient-specific scaffolds, okay, to engineer tissues. So again, this is part of the right coronary artery tree. The coronary artery feeds blood to the heart, and the coronary artery is what becomes blocked during a heart attack, okay? So that would be up here, okay? And so we can make these out of these ECM proteins, things like fibrin, hyaluronic acid, alginate, and collagen. We can have a solid walls and open lumen. It takes about an hour to make this, which is within the viability period of cells if we integrate them. And right now, this has all been done on a MakerBot, okay? This technology will only improve with a higher quality and higher end hardware. And this is the way we actually seed cells on the interior of these arteries to build a functioning artery. So we're not at the point where we can tissue engineer a functional artery for transplant. We've really just developed the technology to start to build these. And there's been a number of research teams and companies trying to tissue engineer an artery for a long time for coronary artery bypass. There's a lot of challenges to doing this beyond just building the scaffold. But what our technology does is allow us to build complex scaffolds, and that's new. So the current techniques out there just build a straight tube. We can actually recreate a lot of the complex architecture in the real coronary artery tree and builds essentially a better potential graft by doing that. So this is just one application. The next one I want to talk to you about is building the actual heart. Obviously a little bit more ambitious than just the artery. But the idea is similar. So in this case, we're not building an adult heart. An adult heart is really large. Right now, we're starting small. We're building the embryonic heart. All right? The embryonic heart is actually the time, in the human at least, that heart muscle forms. So the heart muscle only forms in the human during this embryonic development period. That's the only time the cells in your body have the potential to form new heart muscle. And that's really why we've been focusing on the embryonic state, because we know that in the human at least, that's a potential mechanism or a potential system that will work. To 3D print something accurate, you need accurate data input into the system, right? And this shows our ability to image through an embryonic heart, right? And so what you're seeing, this is the whole heart, these are the ventricles, and this is the trabeculated tissue inside, okay? So we can image the heart in exquisite detail. And then what we do is we generate a 3D computer model from that imaging data. So in this case, you may have noticed that the actual heart had a lot of these internal trabeculations. At this point, we're using a simpler representation of that with fewer internal details. So here's a slice of our 3D printed scaffold that we just overlaid on this internal structure to show that it matches up. And then this next image shows the actual 3D printed heart next to a penny just for reference, okay? So this is a 3D printed embryonic heart. With a lot of that internal structure, hopefully you can appreciate that a lot of these internal features, which might be a little bit hard to see, match up very well with this 3D model. And so our future work then is actually building a functional human heart muscle for both drug discovery and heart repair applications. So that really highlights what we've been able to do recently in this 3D bioprinting space, taking soft materials and using them to really engineer complex structures that were not previously possible, okay? Really biomimetic structures, taking both MRI data and this 3D multi-photon imaging of embryonic organs. So what is the future work and impact, right? Where can this take us? Where are we going with this? So first of all, I just want to show you an example of some 3D printed heart muscle tissue. Hopefully you can appreciate that's contracting. So what we can do is we can integrate cells and start to 3D print actual functioning heart muscle. The first thing we're looking at is engineering human heart muscle for drug discovery applications, okay? To replace or augment or improve what we can do just with animal models currently, right? So a lot of drugs fail at the stage of human clinical trials, okay, because they have cardiac toxicity, all right? You don't really know that until you test it in a human, right? And a rat or a mouse does not really predict very well what happens in the human heart. The second is to enable personalized medicine, right? So you can imagine that a drug may cause an issue in my heart or one of your hearts, right? But not everyone's, right? So only a subset of the population is going to be affected by the negative side effects of a drug. So if we have a patient-specific heart surrogate that we can engineer, you can test a drug on that surrogate and determine if it's a good option for that patient, okay? A third option is to actually engineer heart muscle for heart repair. So right now, these little embryonic hearts that we're trying to make is not going to repair a large defect in an adult, but we're really just at a starting point. And our ultimate vision is to extend and improve this technology so that we're engineering large pieces of heart tissue. If we do that, then we have a large enough piece of heart tissue to repair something like a myocardial infarct for someone who's had a heart attack. That would be in lieu of a heart transplant, okay? So heart transplants are an effective way to treat a failing heart, but donor hearts are severely limited in supply. There's only about 2,000 available each year, okay? And there's at least 40 to 100,000 people that need them each year, all right? So that's the need we're looking to address. So none of this would be possible without the people doing the work and the funding sources to do that. So I'd like to draw attention just to a couple students, Quentin Jallara, John Zemansky, and TJ Hidden, that have led the cardiac tissue engineering and 3D bioprinting work, and then also the financial resources from funding agencies that have sponsored this, including the NIH, the American Heart Association, the National Science Foundation, and the Human Frontier Science Program.